Hi everyone, my name is Ícaro. I'm a doctoral student in the bioinformatics course
Hi.
at the University of São Paulo from Brazil. And today I'm
Okay.
going to present a research project that I'm currently working on in my doctorate with the title Integrative Approach Identifies Host-Microbe Interactions in the Blood of SEPTIC Patients. Well, to bring an introduction part, to date, we know that several studies have demonstrated a synergistic relationship between our immune system and the community of microbes that live in our body, well known as the microbiome. And these microbial communities not only contribute to the host defense against infections, but also modulate the immune response in different locations and tissues. However, under certain conditions that compromise the host defense, some of these species can invade tissues and establish infections. Sepsis, for example, is a condition that occurs due to a strong immune response to an infection. And this imbalanced immune response can lead to organ dysfunction and, in more severe cases, leading even to death. And nowadays, RNA-seq has become one of the main tools applied by researchers for understanding the immune response in infectious diseases. For sepsis is a good example of these applicants. And regarding the RNA-seq data analysis, most human transcriptomic studies evaluate gene expression mainly from the human host. And however, during the read mapping process, most bioinformatic pipelines end up discarding these reads not mapped to the human host. And these map reads can provide valuable information of RNA transcripts, which can be uh, uh, derived from microbes such as viruses, bacteria, and archaea, for example, which can be present in the samples. And then the question is, what if we take into account this information from mapped reads? And that's basically what we have done. So here in this project, we proposed basically a bioinformatic approach to reutilize this biological discarded data of these mapped reads in RNA-seq studies, and we try to generate new insights into host microbe interaction during infectious diseases. As a specific object, we want to identify the potential microbial derived transcripts in the circulating blood of septic patients. And another objective is to identify the differentially abundant microbes in sepsis and what is their contribution in host and immune response, or what is the effect of the presence of these microbes in the host immune response. So for the method section, we divided this in three parts. Basically, the first part is the study selection, which we selected studies from the literature and public available studies, and we selected some studies to reanalyze and apply the bioinformatic workflow that we are working on. The second part was to develop the bioinformatics workflow, and this bioinformatics workflow was made to take into account this information from mapper reads. And then after concluding the construction of this bioinformatics workflow, we apply this approach to the sepsis studies that we have selected. So regarding the selection of these public available RNA-seq studies, we are searching in gene expression omnibus for studies, RNA-seq studies of sepsis, and we found 77 studies related to sepsis. After a manual curation step, we selected three different data sets of sepsis to reanalyze and apply our computation approach. Regarding the bioinformatics workflow, basically our pipeline used this information from these map reads and perform a taxonomy classification to identify the potential microbes that can be present in the samples. So basically, after this taxonomy classification approach, we conducted two more steps of filtering, basically to remove spurious association or spurious classifications, and as well to remove potential contaminant sequence that can be present in the blood. And after this step, the remaining species, we use it as a new layer of information and combining this with the host transcriptome to try to understand the related host microbe interactions. that can be associated with these two different data sets. In terms of results of discussion, first of all, we compared our results. So we have done an experimental validation when we compare our results with the confirmed etiological agents that were found by different techniques in patients who have sepsis. So one of the techniques, for example, is real-time PCR, and the other one is blood culture. And the original authors performed these experiments in the specific studies. So we reanalyzed the studies, and we found the bacteria which were present in RNA-seq data, and we were able to identify the confirmed etiological agents that are shared between which were present as well in the blood culture and the real-time PCR. And as well, we were able to identify some etiological agents that were not found by these wet lab techniques. So this was a really interesting result for us. Going on, after that, we have found the microbes present in this RNA-seq data. We performed a differential abundance analysis, basically to compare the septic patients against their control groups for each of studies separately. And we found differentially abundant species which were shared in more than one studies and are represented here in this network as orange nodes. And the green nodes are the nodes related to studies, and the yellow nodes are represented to differentially abundant species that were found specifically in one study in particular. Based on the previous results of the differentially abundant species, we selected two species, and we performed a differential gene expression analysis in the host now. And we compared the septic patients which have the presence, and this comparison as well was done basically comparing the presence against absence of this bacterial species separately for each of these studies. And doing this approach, we found some genes that were differentially expressed and in each of these studies, and some of them were shared between both of the studies, as we can see in this figure here. In addition, using the gene set enrichment analysis, we identified functional blood modules alterations in the human host by the presence of these bacteria, and also we were able to find blood and transcription modules which were altered and shared between both of these studies, as we are putting an example for here. Well, for the next steps, now we are planning to keep working on in this approach and try to investigate the host transcriptome alterations by the presence of other microbes that we have done before. So basically, we found these effects for 2 species. Now we want to include more species and see what are the effects of this particular of the presence or absence of this particular species. And after that, we want to apply our computational approach in other disease models as well. So related to the potential contributions of this project, we have some of them to list here. First, we developed a new computation approach to identify the microbial community present in RNA-seq data. And after that, we can say that our study can enlighten the potential of reanalyzing the public available data to generate new insights. And last, this research can offer a new perspective to study host microbe crosstalk and their roles in health and disease. These are the references that I use in this presentation from here, and I would like to thank the research funding, that is FOPESP. And I welcome you guys to feel free to enter in contact with us. And as well, I would like to thank you all the guys from my lab. I would like to thank my advisor, Elder Nakaya, my co-advisor, Paulo Moral, and the postdocs, Marielton and Yuvika, that are helping me a lot to improve in this project. So see you guys. See you in the event. Bye-bye.
Thank you, Ícaro. Any questions? Oops. Hi, I have one again. Hi, Ícaro. I would like to ask you, how do you think your approach would be useful in the treatment of the sepsis? You know, this is an important disease that almost require a real-time decision. How do you think you can apply your results?
Hi, thank you for your question. So I think there are several applications for this new study that we are trying to working on, but I think one of the main of these approaches is to try to identify new etiological agents that can be present in the RNA-seq data, and this brings us the opportunity for understanding what is the impact of these particular microbes or even viruses, if we can think about sepsis in a immune repression state. So we can try to understand what is the response of those viruses that are present on the samples, and try to track down and see which patterns we can identify, even in terms of the immune response of these patients. So I think this is the new enlightened that we can see in our research for now.
Okay.